A'udhu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. Dear students of 12th class, subject Botany, Assalamu alaikum wa rahmatullahi wa barakatuhu. Dear students, you know that yesterday we have discussed the last question of the unit first, and the question was outbreeding devices. Dear students, we will continue this question outbreeding devices, and you know that self-incompatibility is the main outbreeding device in angiosperms, in which carpel of a flower inhibits the germination of pollen grain and check the further growth of pollen tube so as to avoid inbreeding and help outbreeding. This self-incompatibility is achieved by the interaction of chemical substances produced by the male gametophyte and the tissue of the style. Dear students, we have discussed it in our last lecture, yesterday's lecture, and as it was our topic yesterday, and we have also seen that the sexual incompatibility is also shown by some species of plants in which the pistil is pollinated by the right type of pollen but fails to set seeds due to inability of their gametes to fuse with each other. And the sexual incompatibility between the individuals of different species is called interspecific, and between the individuals of same species as intraspecific. And we have also seen that the self-incompatibility is controlled by the genes and involves many complex physiological, biochemical, and morphological mechanisms with the interaction between pollen and stigma. Now, we will continue this outbreeding device as it is also brought about by the self-incompatibility, and this self-incompatibility has been divided into two major groups. So our today's topic is types of self-incompatibility. The self-incompatibility has been divided into two major groups. One is called the heteromorphic system and the other is called the homomorphic system. Regarding the heteromorphic system, in this type, same species produces more than one morphologic flowers, and such flowers can be recognized without a breeding test. Now, regarding the homomorphic system, in this type, two or more mating type flowers are of same morphological type and can only be recognized by breeding test. This is the main difference between heteromorphic and homomorphic systems of self-incompatibility. Now, the incompatibility may be due to genotype of plant in which it is located. It should also be mentioned here, and you should understand this, that the self-incompatibility may be due to genotype of plant in which it is located. Now, the biological significance of self-incompatibility. The biological significance of self-incompatibility may be due to number one, pollen stigma interaction, number second, pollen tube ovule interaction. These are the two factors on which the biological this significance of self-incompatibility is depends. Now, in self-incompatibility, fruit trees, in self-incompatibility fruit trees, it is necessary to plant two cross-compatible varieties to ensure fruitfulness. In self-incompatibility fruit trees, it is necessary to plant two cross-compatible varieties to ensure fruitfulness. It means that if in fruit trees self-incompatibility is found, then it is necessary to plant two cross-compatible varieties so that the fruitfulness can be achieved. It can also be used in hybrid seed production. It means that self-incompatibility can be used also in hybrid seed production. This is the one more biological significance of self-incompatibility. Now, lastly, it brings reproductive isolation and helps in emergence of new species of plants. Means that new species of plants are formed by the self-incompatibility. So this was all about today's lecture. So in this way we have completed the outbreeding devices today, and now we have also ended our unit first, and now in our next lecture tomorrow inshallah we will start a new unit and we'll see the different types of questions which have been included in that unit. And till tomorrow, stay home, stay safe. Thank you very much.